The mysteries and paths of Alzheimer's disease. Subjective memory complaints and a decline of performance of daily activities may indicate early stages of Alzheimer's disease, or AD, particularly in patients aged 65 or older. AD is a progressive disease, beginning with pathological changes that occur decades before clinical symptoms, leading eventually to the development of mild cognitive impairment, functional symptoms, and then dementia. AD accounts for as many as 80% of all dementia diagnoses. Today, we are able to diagnose AD in its early symptomatic stages by assessing biomarkers and by using tests to evaluate cognitive performance, functional impairment, and behavioral symptoms. To better understand the disease, we must look back in time to the pathological origins of AD within the brain. The two pathological hallmarks of AD appear at distinct locations within the brain. Plaques made up of A-beta occur outside of neurons, while neurofibrillary tangles composed of tau protein develop within neurons. Abnormal accumulation of A-beta within the brain may begin decades before symptoms appear. Amyloid beta is produced when amyloid precursor protein, or APP, is cleaved by beta secretase, base 1, and then by gamma secretase. A-beta is formed in the course of normal brain metabolism. However, it may be overproduced in certain individuals. In healthy people, A-beta is cleared from the brain through systemic circulation and local processes such as uptake by microglia. Both the overproduction of A-beta and inadequate clearance mechanisms can cause A-beta peptides to accumulate in the brain and self-aggregate into plaques. Plaque deposition is first seen in the brain's neocortex, the center of higher mental functions. It progresses to the more primitive cortex, including the hippocampus and related structures involved in learning and memory. Finally, it spreads to subcortical areas that regulate attention, emotion, and various other activities. Positron emission tomography can detect evidence of A-beta deposition and plaques. Low levels of A-beta in cerebrospinal fluid, or CSF, can also indicate pathology. Over time, the accumulation of amyloid plaques activates cells of the neuronal immune system, potentially causing a chronic inflammatory reaction that has toxic effects on neurons. A-beta may also disrupt the energy supply of neurons and induce oxidative stress, causing further cellular injury. Tau protein pathology begins to appear approximately 15 years before the onset of clinical AD symptoms. Tau is normally associated with microtubules, which maintain structural and transport systems within neurons. In people with AD, the presence of A-beta may promote the abnormal phosphorylation of tau, followed by its detachment of microtubules. Tau subsequently associates into insoluble structures called paired helical filaments, which further aggregate to form neurofibrillary tangles. Higher abnormal levels of tau can be detected in the cerebrospinal fluid. While neurofibrillary tangles may appear in the brains of people with other neurodegenerative diseases, there is growing evidence that the toxic effects of tau and A-beta may amplify each other in people with AD. Eventually, the underlying pathology of AD causes synaptic dysfunction and neuronal loss. This results in cortical thinning or atrophy, leading to more advanced symptoms. As the disease progresses, it extends to different parts of the brain, with corresponding biomarker evidence of pathology. As more regions are affected, more symptoms appear. Over time, pathology spreads to regions of the brain that control basic functions, such as heart rate, leading ultimately to death. AD is often diagnosed in the mild AD dementia stage, and average survival is 4 to 8 years after the diagnosis of AD, but can be longer depending on other factors. With ongoing advances in AD research, we continue to enhance our understanding of underlying disease processes and the sequence in which they occur. We now know that AD begins with clinically silent processes, which lead to the appearance of symptoms decades later. Diagnosis of the earliest stages of AD is being aided by emerging risk factor evidence, improved screening tools that evaluate symptoms with greater accuracy, and novel techniques for the detection and measurement of AD biomarkers. It is hoped that ongoing and future research continues to help unravel the causes of AD and hopefully aids in better treatment of this complex disease.